Can you hear me? Okay. This section, we want to speak about bacterial genetics, which is we speak about peptidoglycan membrane and organism of bacteria, but not about genetics. What is genetics and how it makes this role? Okay, you know, prokaryote and eukaryote genetics. Do you know what's the difference of prokaryote and eukaryote genetics? Do you know? Can you explain for me only one or two different? If you don't know, please to me, I continue. But if you know, please say. Okay. Do you know different of genome prokaryote or eukaryote? Do you know? Okay. Is it okay? Is it okay? Hope. Do you know different of eukaryote and prokaryote genomes? And I want teaching you. Yes. Nuclei. Eukaryote have nuclei, but prokaryote don't have. Yes. Eukaryote have, but eukaryote don't have. Prokaryotes are circular. They have circular DNA. But what? What? Eukaryotes have a strand DNA. You know. And another prokaryote are N, but eukaryote are 2N, means haploid and diploid. Okay. So, so prokaryotes are haploid and they contain one single circular chromosome. And prokaryotes often contain a small circular DNA molecule called plasmid that contains loose prokaryotes such as circular DNA molecules. Prokaryotes replicate. Okay, it is right. But eukaryotes are diploid, have linear chromosomes, many more than one. Diploid prokaryotes are diploid. Prokaryote is single cellular. Eukaryote is more than one but linear chromosome. Write them and then go another slide. Yes, I will send you, but now please write and listen, Malva. Don't worry, I will help you, I will send you. I really taught you. Thank you. When you write, tell me, I continue. Okay. No, sound is okay. I'm here. Let's go another. In eukaryote, transcription of gene is in RNA occurs in the nucleus. Yes, you know. and translate RNA to protein occurs in cytoplasm. Because eukaryote have nucleus, but in eukaryote, 2% of separate of both of them, I mean transcription and uh, translation occurs in cytoplasm. This is another. In eukaryote, all of them occurs in eukaryote. Transcription in nucleus, translation in cytoplasm, but in prokaryote, both of them occurs in cytoplasm. Please write, and I will say. Definition. Okay. What is definition? Plasmid. A small circular DNA molecule with easy manipulation. They are usually transmissible with continuous replication. Plasmid is a small circular DNA molecules which have easy manipulation. They are usually self-transmitted with spontaneous replication. Please write this slide. Do you hear me? What is replicon? DNA or plasmid replication information. We call replicon. The genes are basic from bacteria are carrying on this chromosomal DNA, and the genes are plasmid gene for special purpose. Please pay attention, special, not essential. It's like this. Okay. Transposons. What means transposons? They are mutation. You know mutation? But insertion mutation. Such mutation we insert in DNA. We call them transposition. Do you know in English transposition, in Persian transposition? Excuse me, do you hear me? Why you don't pay attention in class? Why? No sound? Okay. Please wait. You hear me? That's okay. That's okay. That's right. No, yes. Oh, now yes. Okay. It's important header. Okay, transposition is insertion sequence which carry the transitional gene. In IS, we have transitional gene. Complex transposons carry the other genes like AB residence. The complex transposons are included the IS transposon. and they can be despite of plasmid or the lack of replication information they are. The replication is related to replicon on Z. Okay. Bacterial genetics. What is this bacterial genetics? Acquiring gene through gene transfer provide new genetic information to microorganisms. They changing with environment. The major source of variation are mutation. You know mutation? What is mutation? In mutation, usually only in single gene change at any one time. In contrast, gene transfer results in many genes being transferred simultaneously, giving the recipient cell much more additional genetic information. Okay, if you have any question, please ask me. I can explain more. You understand? Okay. And bacteria are haploid. I told you, bacteria are haploid. Eukaryotes are diploid. It means they have only one DNA. Bacteria don't have sex like animal or human or plant. They have belief, something like sex. And bacteria acquire DNA from other bacteria. It's like they don't have the sex, but how they provide their DNA from another bacteria? How? With this transformation, construction, and conclusion. This DNA may or may not recombine into their precise genome. And we use phrases like lateral or horizontal gene transfer to describe this sexual. Like this slide, it is important for you. What is transcription, transduction, or conclusion? Do you know? Do you studied before? Please write. or some of me now. What do you mean, Fatima? Okay. Bacteria DNA means bacteria which are mutation. There will be damage because mutation and natural selection selected. Okay, let's go next slide. This is transformation. You know, transformation is simple. It's simple way of genetic in bacteria. In transformation, here, look here. Okay, right here. Okay, I know. Okay, transformation. In transformation, what happens? Look here, in transformation. This is one DNA which integration of signal strand donor DNA by breaking. From this slide, please look here. This is one bacteria, and this is DNA from another bacteria. Okay, do you understand? We want to insert this DNA to our bacteria and have mutation of our DNA with foreign DNA or another DNA. Do you understand? Look here. Only Fatima Adnan is here. Afia, Asra, Samer, Habib, Hamze, Roya, Ehsan, Ahmad, Ali Hussein, Zulfaqar, Abdul Hussein. Where are you? Okay. Hello, my friend. I think I should call you. Okay. Listen to me. Transformation means another DNA from another bacteria insert into our DNA. Okay. We have this DNA from another cell we call, do you know what we call? Donor. You know? Do you understand? Do you hear before? Donor. Okay. DNA from donor bacteria. We have bacteria we call recipient DNA. Single strand of donor DNA enters to our cell and go to our DNA. This is like integration of single strand donor DNA by breakage and reunion in our cell. Donor integration to our previous DNA. It make mutation or mutation. We have new DNA with mutation. You know, these two strands open. One of them is natural, and one of them have another DNA from donor DNA. We call this new mutation or new mutation bacteria, and it is like previous, like natural, like mother. Yes or no? Do you understand? Please write this. Please write this. Please write this. They are very important. I'll ask you please explain for me transformation with picture. Please write. Okay. It means, yes, very important, Fatima. It means, this, yes, donor means the bacteria which give part of its DNA. Parts of its DNA, you know, it is donor cell, and which accept this part of, we call them recipient cell. Do you understand, Hassan? Yes, it's important. Please write for yourself. I wait for you. When you write, tell me, go to next. Okay, and write this, please. Okay, and write this, you. Okay, next. Okay, there are some natural components in bacteria. They have transformation. Means which bacteria? Bacillus subtilis, Neisseria gonorrhea, Streptococcus pneumoniae, Haemophilus influenza. They have transformation. Here, transformation, one exam transformation. This is the first exam for explain transformation in this year, 128 with Griffith. This is the natural cell, and this is a living cell, but without um, capsule. What which bacteria is? Which bacteria is? You know, this is what bacteria. Which bacteria is? Which bacteria is? Please say which bacteria is? Which bacteria is? You know, this is Streptococcus pneumoniae. Pneumonia. It causes pneumonia. Pneumonia, Streptococcus pneumonia or pneumococcus. It causes pneumonia. If pneumonia, do you understand, Rafaqar, what is this? If pneumococcus, we have pneumococcus and have capsule, it was immunogen and make disease. If you have bacteria with capsule but not living bacteria which hit like but have capsule for calibration. And with living non-capsule, we insert most will be die because okay, have some rest and we start transduction. Yes, yes, yes. Okay, let's continue. Let's continue. Okay, what is this? Okay, for transduction, we need for transduction, we need phage. Phage are dialytic or porophage phase. What is this? We have lytic, we call them lysogenetic phage or porophage. We have two kinds of phage, lysogenetic and porophage. Lysogenetic bacteria are the bacteria carry the phage which bestow a new character to them, like lysogenetic dehydroesterase can produce toxin. Excuse me. And what is phage? Prophage means temperate phage can enter to prophage phase like lambda-phage. You should learn this. Lambda-phage for prophage and lysogenetic dihydria for lysogenic bacteria. Please write this. Please write. Okay. Fudge. This is fudge. This is fudge. What is this here? You know this. What is this? Do you know? Between two or their function are different. Their function. Please look here. Please look here. Fatima, your question is good. Please, lysogenetic means the phage beat lysis. Okay. Phage. Lytic phage in lysogenetic phage are T11. We call them T2 or T4 in E. coli. And temperate phage like phage lambda and filamentous phage like M13 phage. Right? This kind of phage. We have three kind of phage. Okay. Note, temperate phage temperate phage can enter to lytic phage, and bacterial DNA can enter to filamentous phage like N30. These linear DNA are used in sequencing and genetic manipulation. Right? I want to continue. Okay. This is bacteriophage. This is transduction. Excuse me. This is transduction. Transduction occur with bacteriophage. This is bacteriophage. Phage. This red is DNA. Phage DNA. Phage DNA. Look here. Red is phage DNA. Here. Look. One phage with DNA attached to bacteria, new bacteria, and enter his phage DNA to cell. Then DNA infect the host cell, and it may cause the degradation of host DNA into a small fragment. Then phage coat proteins synthesis means this synthesis for this DNA with DNA replication, and then during mutilation of virus particles, a few phage heads may envelop fragments of bacterial DNA inside phage DNA. Only bacterial DNA is present in transduction variant. And finally, the new phage with DNA infected another cell and transferred the bacterial DNA into the new cell. Okay, when you write, please say me. Other organisms, no. Other bacteria, they can infection other bacteria only. Okay, you don't have any question more? We should make it smaller. Write this and I explain right. Okay, we have some new cell with bacteriophage and DNA, you know? Yes. During the maturation of virus particles, a few phage heads may involve fragments of bacteria DNA inside the phage DNA. Only bacteria DNA is present in the transduction period, and they go in outside the cell and can infect another cell like this. Neophage with DNA of neohost like this, okay, can come to another cell and make another cell infect, and it can go to DNA house, replicate on this, and you have two cells with two new DNA or two new modification like this. Okay, we want to speak about conclusion. Conclusion. Write this slide. I got discount. Do you hear me? Do you hear me? Do you hear me? Okay. What is conclusion? Conclusion means transfer teragen in conclusion, like what we have sex pill here, like we call a factor. Plasmid usually transfer by their teragen in conclusion. F factor produce sex pili. We have sex pili, we call F factor. HFR bacteria, if plasmid or fertility factor integrate in the bacteria chromosome, the HFR bacteria will be produced. They have HFR. We call HFR plasmid to plasmid which have integral bacteria chromosome, which receive bacteria, or it may female bacteria. Yes, male is F factor, female is HF factor, or HF factor is high frequency recombinant. R factor can transfer during conclusion. Okay, you understand? Like this. Here, look here, look here. This is sexually, okay, and it can be F factor and it can be HFR. Look here. Okay. Do you have any question? Okay. This slide is very good for you and explain enough to you. This is F factor. F factor. Donor cell F positive. You know? It is cell with pili, sex pili, and it has F plasmid, which one transfer this DNA to a new cell or new bacteria which haven't any plasmid. We call F negative. Do you understand? Here, please look here. Do you understand? Okay. Conclusion start. We have one cell with sex pili and plasmid. We call F positive. And we have one cell without plasmid and pili. We call F negative. The conclusion: sex acquire between them, pili attach F negative cell, and they be together, and plasmid transfer to new cell, and then this plasmid make our cell F negative. F positive cell, it make our cell, which was F negative, make F positive cell with pili and plasmid. End of this, inside of one F positive and one F negative, we have two F positive cells. Please write this. This is important. I explain you more. Whatever I will explain you, please write this. This is important. Rena. What move? What? If negative bacteria doesn't have plasmid, we transfer with P6 plasmid. If cellule was F negative, what it can transfer? What? Do you understand? Okay. When you draw, tell me I continue. Okay, let's next slide. Conclusion continue. Okay, look here. Conclusion continued. Okay, we have F cell, positive cell. Recombination between F factor and chromosome occurs at the last slide. And it means if in bacteria, in chromosome of bacteria, this is chromosome of bacteria which receive DNA, the donor DNA. They insert plasmid factor inside of chromosome. We have this. We call this HFR. We call this HFR. Draw, please. Draw. Okay. Next slide. Here we have HFR cell which has one part of plasmid of another cell, and we have one F negative cell. The integration, and we have two cells. One of them with chromosome of bacteria, and one of them with new plasmid chromosome. Okay. Okay. These are good. These are useful. Please write this. Please write down this. Please down this. It's hard, yes, I know. What if you watch the picture or looking at picture, it maybe make, yes, I know you are tired, but some more, we should continue. Okay, that's finish. If everyone have question, please ask me. For next section, please remember me. We should study about bacterial metabolism. Okay, Marve, can you please remember me bacterial metabolism? Okay, okay. Haydar, Qasem, Fatima, Adnan, Marve. They are very active in class. I remember their name when I want to give them number or give them point. Okay? Thank you, mom. Please remember me uh, here, Marve. Please remember me. You're welcome, Fatima, Adnan. Thank you, Rena. You are active too. Thanks, Roya. Thanks a lot for your attention.